Welcome to Basic Genetics course. I am Jagat Kumar Roy. I am a retired teacher from Department of Zoology, Banaras Hindu University, Varanasi. There are other two teachers in this course, Dr. Richa Are and Professor Madhu Gavaldas Tapadia. This is the third lecture of week 8 and the title is Anomalies in Chromosome Structure. In earlier lecture, we talked about anomalies in chromosome number. Now we are discussing about chromosome structural anomalies. Once again, I show the same slide, that is how the chromosome look like, and the chromosome contains DNA. That is what? Or chromosome contains genes. When we say chromosome contains DNA, different segments, that is one stretch of nucleotide pair, constitute a gene which gives one mRNA. So, when we say about the structural anomaly in chromosome, that means if some part is deleted, if some part is transported to other region, the genes are actually getting shuffled. So, chromosomal anomalies, an abnormal number, that is numerical anomaly, or an abnormal structure, structural anomaly. Today we will focus on structural anomalies, and structural anomalies can be of 4 types: deletion, duplication, inversion and translocation. Chromosomal mutation are changes in chromosomes. I am showing here the deletion, that is, you can see this chromosome. So, this is the chromosome, and in this chromosome there are 2 cuts, 2 breaks. Such kind of break can occur when high energy radiation, these chromosomes are exposed to high energy radiation. Every day we are exposed to certain kind of radiation, and such kind of breakages are possible. If suppose this broken part is removed, has come out from here, and the other part of the chromosome, this part and this part get joined together, then this region is lost. Whenever there is chromosome break, immediately it is natural that repair process will be on and chromosome will get joined. In the process of joining, if this part is shifted, result is this top part and this bottom part getting joined. So, this is how the deletion occurs. Now, we take some example of deletions, whether in nature such kind of deletions we find. The example is cry of cat, or the actual name is cry-do-chat syndrome. This is associated with deletion of short arm of chromosome 5. This is chromosome 5. This is chromosome 5. This chromosome has 2 arms because this is the centromere. From centromere, this is one arm. This is another arm. Now, the short arm, you call it as petite arm or P arm. The long arm, you call it as Q. The petite means small, and that is how the small arm is called as P. The Q has been used because the short arm has been considered as P. This Q is nothing to do with something else. So, here in cry of cat syndrome, a part of the short arm is not present. So, what kind of this occurrence? This is in 1 in 20,000 to 50,000 live births. The symptoms are high-pitched, cat-like cry of the baby or weak cry. This is due to muscle weakness. They meow like cat. They cannot cry loudly. Low birth weight, small head, rounded face, broad flattened bridge of nose, that is flat nose, eyes spaced wide apart, folds of skin over eyelid, this is called as epicanthal fold that I had talked in earlier lecture in Down syndrome also. Then abnormalities in palate, such as an usually narrow and high palate. So, the mouth is narrow, palate is quite high. So, this is one example of the deletion. Another example of deletion is from tumor suppressor gene RB. This RB name has come from retinoblastoma. You can see here, this child is having tumor in the eye. This is the tumor of retina and this is called as retinoblastoma. If the eye is not removed, this child will have cancer spread and finally this will die. How this kind of cancer occurs or tumor occurs? If RB gene is deleted. Where is RB gene located? This is in Q arm of chromosome 13. And here, what this gene does, why there is cancer? This gene has a very important function. One calls it as a tumor suppressor function because this does not allow cell division to occur. This is the picture of a cell cycle. This is cell cycle, where a cell after division comes to G1, that is growth 1, where it prepares for DNA replication. Then it undergoes DNA replication. After DNA replication, there is growth 2 or G2, where it prepares for the mitosis, that is cell division. So, this RB gene product, that is RB protein, what it does, that in G1, this binds with one of the important transcription factor E2F and does not allow this to function. This E2F, this is one of the transcription factor which is very important for groups of transcriptions or groups of gene activities which are essential for G1 and which is essential so that this will go for S phase. But if RB, this is RB protein and this is holding this. So, result is this cell cannot go to S phase. That is how this cell cannot divide. If this, that means under this condition no cell can divide, but under certain condition a cell divides. And how cell divides? That this RB is phosphorylated, phosphate groups are added on this RB. Result is, for a time being, this leaves E2F and thus all preparations are done, and after that this again comes back and holds the E2F. So, only one cycle of replication is allowed by this phosphorylation. But if this RB is not there, if there is a deletion, result is the cell will continue its division one after the another because this RB is not present, this is gone. So, E2F is free and this will allow the preparation to occur again and again as it comes in G1, and cells will divide once after another like that. So this is another example of deletion. Now let us come to duplication. Similarly, like deletion can occur, duplication can also occur. Here in this picture, this is shown that this is a part of chromosome. In the second picture, this is shown this part is duplicated. Such duplication can occur if there is pairing of the two homologous chromosome not matching each point, but if the pairing is abnormal, this can result in one chromosome having part of another chromosome, and that is how a duplication can occur. The example is Charcot-Marie-Tooth disease type 1A. This is caused by duplication of PMP22 gene. This is present on chromosome 17. This is one example. The other example is Pallister-Killian syndrome. This is resulting from duplication of chromosome 12. So this is another syndrome where the duplication is involved. What happens with duplication? That means one gene copy is additional. So that is causing the problem. Exactly in the same way, the inversion can occur. We were talking about the deletion. That is, this is a chromosome. This is a chromosome. Here there is one break here, another break here. Result is this part could have been deleted, but because the cell always tries to repair or always try to join the broken part of the chromosome, so this can join this also. If it is joined normally, then the chromosome is normal again. If by chance after the cut or after the breakage, if this inverts and gets joined, now this is inversion. So this is an inversion, example of an inversion. In this inversion, the centromere is not involved. This is the centromeric part. Here, the centromere is not involved. So such inversions are called as paracentric, means not involving centromere. The other case, breakages occur in such a way that centromere is in the middle. And now, if this inverted and getting associated with this, then this is called pericentric inversion. Pericentric means the inversion around the centromere. We will talk about the paracentric inversion as well as pericentric inversion. It's means what kind of significance it has, and we will talk about later. But at this moment, we are talking about how the structural anomalies can occur. The fourth one is the translocation. In this case, this is one chromosome, this is another chromosome. They are non-homologous chromosomes. These are not the two homologous chromosomes. If breakage occurs in one and breakage occur in the other chromosome, and if the two parts can get joined, then the new chromosome will be like this. See, this is the chromosome A, this is chromosome B, and now this chromosome B part has come to this B part has come to this. So this is the chromosome which is now longer. The other one has got the short part of it. This is the chromosome which is shorter. So I give you one example of this translocation. It is written like this, T in parenthesis, if the translocation in between chromosome 9 and chromosome 22. Here chromosome 9 is involved, this is chromosome 9 and chromosome 22. These two chromosomes are involved. So, the T9 semicolon 22, that is how it is written. This translocation, when such kind of translocation occurs, that a part of this chromosome is going here and a small part of this chromosome, from this large chromosome, chromosome 9, a small part come here and a large part of chromosome 22 is going there. Result is, this chromosome has become now longer, and that is why now this chromosome is written as in chromosome 9q plus. That is, this is P arm, this is Q arm. The Q arm is now longer. So that is why this is called as Q plus. And here what has happened? That 22q, this is 22q minus, because this chromosome is shorter than this. What important thing has happened, that the two genes, a gene called BCR, which was here, and a gene called ABL, which was here. So this is ABL and this is BCR. These two genes, they have come together. So now you have BCR and ABL, they are together. BCR name has come from breakpoint cluster region. Earlier this gene, it was not known that this is a gene, but in this region, lot of chromosome breakages were there. So that is why breakpoints were earlier known for this region. Later this was found that this codes for a protein also. So that is how this is now a gene. Now what has happened? BCR is fused with ABL. ABL is it codes for a tyrosine kinase, which is very important for cell division. And if tyrosine kinase gets deregulated, that is, if its production is more, result is cell division will be much more, and this can lead to cancer. ABL gene is very well regulated. It is such regulated, it has a lot of regulatory region, and they are regulated by various factors. Now when ABL is fused with BCR, those regulations have gone. As a result, now this makes the cell for division, and that results in cancer. So this results in the cancer named as chronic myelogenous leukemia or acute lymphocytic leukemia. These two kinds of cancer, because of the translocation of these two regions, but in BCR region at the two other points. So that is how these two cancers are there. This is the example of CML or ALL. This is normal blood. In normal blood, RBCs are there and WBCs are there. Fewer WBCs compared to large number of RBCs, and these are platelets. In cancerous condition or in leukemia, these leukocytes, that is the WBCs, they are plenty, and RBCs now comparatively less. So this is a anemia kind of a condition, and this is cancerous. So with this, we come to the discussion. Do we find, the question here I am asking, do we find terminal deletion in chromosomes? We have shown that deletion, the middle part of it is deleted and again rejoined. Is it possible that terminal deletions are also there? If yes, then cite some example. If not, discuss possible reasons. The other question is how the duplications arise. The third, what are the consequences of each type of structural anomalies in meiosis? So with these questions, I stop today. Thank you.